And in this video, I'm going to be presenting a project that I did as part of work at the BioNet lab at Columbia University. So this was part of a larger effort to explore the function of the circadian clock of the fruit fly. And so I'm going to start off by giving some context for how this project, HemiMap, came up. So the circadian clock of the fruit fly is composed of around 150 neurons within the brain of the fruit fly that express so-called clock genes. These clock genes code for a set of proteins that interact in such a way that their concentrations fluctuate about every 24 hours. And so for our study, we were interested in understanding, these neurons, where they're located, um, you know, their morphology, and also how they're connected with each other. And they're broken up into these 7 different groups of neurons with different numbers of neurons within them. And so in order to start digging into, this clock and the neurons within it, we have the HemiBrain dataset, which was released in 2020. And this is the largest synaptic level connectome in existence. It contains the sort of like a skeleton that shows where each neuron is located within the brain, as well as the connectivity between neurons, and you can even it even shows like the like synapses, between neurons and where they're located on the neurons. So it's a really rich piece of data, spanning all the neurons in this blue area of the overall fruit fly brain. And so, for the study, we're hoping to, use this data as a basis to understand the functionality of the circadian clock. And so, here we've, gone through the paper, that they released with this HemiBrain dataset, and they actually went through and labeled a good number of the circadian clock neurons. And so here I've gone through and visualized them in NeuroNLP, which is a tool that is released by the BioNet lab. And, the colors here show different types of circadian neurons. And there's a few, neuron types that are missing from the data set. So this is just kind of showing those neurons in, different colors here. And so, with, excluding the missing neurons, we have about 60 of the projected 75 circadian clock neurons that should exist in just the right hemisphere of the Drosophila brain. If you include the left hemisphere, it would be 150. So just right hemisphere is 75. However, the data that we have from the the HemiBrain data set is, it only contains the connectivity and morphology data from our neurons, and we're interested in function. And that's been studied in, other research done by a number of biologists. And so if we want to be able to study the function and to say like, oh, this neuron in the HemiBrain dataset had this function in this other study, we'd better be sure that the, labels that these studies are giving to these neurons are actually corresponding to the other neurons, right? Just to make sure that we're not, putting the wrong functionality in the wrong places. And so, for example, we have a study done by Professor Fernandez at Barnard, where she was analyzing DN1P neurons. And here are a few of like the microscopy images that she had from it. And so we come to this problem of, we have these labeled HemiBrain neurons as well as existing microscopy data from experiments like Professor Fernandez's. And, we'd like to be able to validate that these are the same types of neurons or the same neurons. And so that's kind of the idea behind HemiMap. So the first thing that we have, to start developing, a way of checking that these are the same is the actual microscopy data that Professor Fernandez provided us on DN1P neurons. And this consists of TIFF files, which are, 5 by 512 by 512 in x and y directions, with 57 z coordinates in two channels. The channels just indicate which dye and the settings of the microscope that were used when they were collecting the data of, where the neuron was positioned in the brain. And then the x, y, and z coordinates basically just indicate different slices that were taken, of where, in the output, and readings in the microscope at different z coordinates. And so, what we have is like a 3D image of the microscopy output, right? And so, we're interested in just the first channel here, because, for this study, we just kind of didn't use the second channel, just used the first channel. And what we're showing here is I went through and took the for each of the x, y coordinates, went down all the z coordinates and just added up all their values to give you the image here. So this is actually representative of a 3D image that would actually have some depth, but instead you're seeing it as just this 2D cross section. And so that's the microscopy data. And a member of our lab, Mehmet, actually developed a tool called HemiMap, and sort of its utility for a certain way of looking through the hemibrain dataset. And so the idea behind it is that you should be able to draw out kind of a white blob like this. And this blob should indicate a portion of the brain, so corresponding to this cross-section here, where you want to find neurons from. And HemiMap will take that in and actually plop out, a bunch of neurons that overlap with this region. The way it actually does that is that HemiBrain contains these pack bits, pieces of images for each neuron. And what this is showing is basically its location in this XY domain, as in space, and the actual color indicates its depth. So the Z coordinate to where it's located. And so HemiMap uses this to actually give you, finds what neurons are actually overlapping with this white portion, and gives you these neurons here. And so, the idea behind HemiMap is actually, we have these microscopy data, so why don't we try and produce one of these, white blobs here, run it through HemiMap, and see if what comes out is actually the DN1P neurons. And so, the way that we're actually going to go about doing this is we actually need to learn how to create a this white blob image from the microscopy data. So for the microscopy data, we have this microscopy reading, this 3D microscopy reading, and this PackBits data. What we're going to do is we're going to pre-process both the microscopy data and the PackBits data. And what these are each representing is, this is representing, so this is a representative of a 3D image where a 0 means there's no DN1P neurons there, and a 1 means there's a DN1P neuron there. And this is the same. This PackBits thing, but is the same, but in 2D. And so our goal would be able to, to actually take the location of the 3D image here and actually map it onto this PackBits representation. But how do we, but for actually doing this, we're gonna represent these images here as point clouds. The way we're going to do that is for each one in those images, we're going to create a point. For all the zeros, we're not creating points. For each one, we're going to create a point. We're going to give it the coordinates of it, just its indices within the image. So each voxel here is assigned to its coordinate. Each pixel here is assigned to its coordinate. So we have these 2 point clouds. And so the idea would be to learn some transform, specifically an affine linear transform, that would just take the coordinates of each of these points and map them to coordinates in 2D space. And then we just try and get the 2 point clouds as close together as possible. And so, the first thing to understand about this process is how the microscopy data is pre-processed. So our goal here is to isolate the voxels that actually contain the neuron, because there is some noise. And so the first step that we take is that this microscopy image doesn't actually indicate where the neuron is, it just has microscopy readings. So we're going to, first off, just set a threshold where voxels that are above that threshold are set to 1, and those below are set to 0. And so we can see, the result of this here. There's a couple sections where it's kind of like faintly, above 0 that are removed by this. And then, you can still see that we have these weird bits that are left over. And so to remove those, we just sort of set everything outside of this red box here, this region of interest, to 0. And so then we get, this image here. And what this image is actually is, it's the 3D image, but I've gone through each of the z-axes and just summed across, all the values. So, the value you get here would just be would just indicate the color would just indicate the number of voxels that are like in that XY axis. And for the PacBets representations, we start off with this figure here. We're just going to remove the color, so just looking at the XY coordinates for each of the DN1P neurons, and then we're going to take each of the DN1P neurons and sort of for each of the XY coordinates, just do a bitwise OR on it, so that we're just, produce an image where having a 1 at a given pixel indicates that at least one of the DN1P neurons exists at that XY coordinate. And then the final step here is kind of motivated by the fact that our OIB channel, or OIB data, only has one hemisphere of the, present. So it doesn't have like, another symmetric half. So we have this weird, bit that's existing in the HemiBrain dataset. And so we're just going to discard that and just look at this red portion here. And so we're just setting everything else to 0, and we get our representation. And so now, we're trying to learn an affine linear transform. And, a simple way of doing that is that a 1-layer neural network with, a weights, a bias, but no activation is actually an affine linear transform. So we can just learn this using gradient descent. However, the loss is a bit more tricky. Recall that we're trying to, match up 2 point clouds as closely as possible, right? Our mapped point cloud and the point cloud for the PacBits data, right? And so there's a lot of work that's done on measuring the distances between point clouds. And so we're going to use one of those distances, the symmetric chamfer distance, to train our network. Now, the chamfer distance and, symmetric chamfer distance, they're trying to capture what's called, the optimal transport distance. So like optimal transport is, a mapping that goes from one set to another set while moving each of the points as minimally as possible, where the as minimally as possible is dependent on like how you define the cost. And so the chamfer distance is trying to capture that. It's like, if I move one of these sets as little as possible, like what would be the cost of like moving it onto the other set, right? So the chamfer distance, not the symmetric one, just the chamfer distance would be this first term here, where you're assuming that the sets are of equal size. So for each of the, points in set 1, you're going to find the closest point in set 2 and add the square of its distance to this whole term here. And so that should kind of capture the distance, between 2 sets if they're of equal size. But this can become a problem if the 2 sets are of different size, which ours are. If you look here, right, this is a lot more points than the 2D. The microscopy point cloud is a lot more points than the packbits point cloud. So by adding this other term, it's just doing the same thing, but with like the two sets reversed. So it solves that issue of scale. And so, putting this all together, we get the following image, which shows the how well our mapping has worked. And so the blue here is our packbits representation, and the red here is the mapping that we've learned from our previous point cloud, where the purple here is the actual overlap. And so what you'll see here is that, what this has done very well is it's learned scaling very well, right? The scale of the red and the blue point clouds are very similar. And it's learned to set the center point very well, right? Like the center of this is very close. But it hasn't learned much about orientation. And so, to try and solve this in a very heuristic way, I took a look back at this, HemiMap, at this microscopy image here, like this cross section that I'd created to kind of show this, and this packbits representation. And if you look, right, if you were to just take this 3D image and just bitwise or down each of the axes to produce a 2D image, you can see that if you just flipped it along that axis, so the vertical axis, and then turn it a little bit, it should be right on top of this other representation, right, with some scaling. And so given that idea, I went ahead and tried mapping that resultant point cloud onto our packbits representation, and we get the following, result, which is actually pretty similar to our previous, image here in terms of the issues with the result, right? It's very well matched in terms of the center, and in terms of the scale, right? But there's obviously an 180-degree turn that needs to be made here. And so, and you can kind of see why it's stuck in this local minimum, because if you were to try and turn this even slightly, it's going to get big losses along the chunk where most of the neuron is located. And so what's happened here is that it's initialized and just moved the neuron and scaled it properly. And so that this is as far as this project has gone. However, possible future work might be taking this existing chamfer distance and adding a term that captures the way the figure is rotated, right? So, for example, you take the center point, which the affine linear transform you should be able to just tease it out of the affine linear transform, and find some way of capturing the difference in angles. So maybe you take each given point and you find the angular distance to the closest point in the other set, right? Just using the center point as the center, and the this point here is the radius, the distance as the radius here, and just find the closest point along. Or you might take a line going from the center point, from this any given point to the center point, and just find the closest point in the blue to the red point you're interested in, and vice versa. Either of those ways might yield something to the loss that actually forces some rotation to be learned. And that's the presentation. Thanks for listening.